I think. Yeah, I think. Okay.
Okay, let's get started. So I just wanted to start off by saying that I'm simultaneously really happy and really sad that Ari is defending her thesis today after spending several years of grinding it out in my lab. Thinking about what to say during this, this is like the most stressful thing that you are asked to do as a PI, is to stand up and try and summarize a person's entire PhD and what it was like to work with them in just a couple of minutes. But I thought that the thing that may be most appropriate in Ari's case is to go back in time and give you a little bit of context for exactly the history of the project that she ended up working on in my lab, how it started, and what the state of things was when she made the decision to join us and work on Danianella cerebrum. The history is, about 10 years ago, when I was finishing up my postdoc, I got to thinking about whether there might be better fish model systems for doing kinds of systems neuroscience questions that I was interested in. And so at some point, probably late at night after a couple of years, I did some Googling to figure out what the world's smallest fish is. It's not Danianella, but they're close. And began thinking that, well, this may be like actually something that we could turn into a laboratory model that may benefit us and other people who are interested in, using zebrafish and related species to understand how the brain works. And that was kind of all that happened with that. It was just a weird idea that I returned to mentally over the next few years, and developed it in the sense that I spent time on online fish hobbyist forums trying to figure out where we get the animals if we were to work on this. But nothing ever really came of it until I somehow miraculously convinced the National Science Foundation to give us some money to work on this. And that's where things got terrifying, because it took this weird little pet idea that I had been nursing for forever and turned it into something that I actually had to do. And that's terrifying in the sense that, it's hard to overstate just how uncertain this was as a project. I think, without question, it is the riskiest thing that my lab has ever done. And so it really took somebody who was either especially naive or especially brave to assume the responsibility of turning this weird idea into something real. And so that's where Ari came in. And I will say that although she was a beginning graduate student at the time, I don't think she was particularly naive. And certainly she was really smart. And even more than that, I think she was especially brave, because she didn't really bat an eye when I brought up the idea of her working on this project for her dissertation. That's great for her. I like to imagine that she wasn't overly worried about the project's likelihood of success in the early stage, but I was terrified. This is also generally true, I think, kind of definitive for my experience as a PI and in mentoring students in particular, that it's really scary to put somebody on this idea, no matter how well you've developed it in your own mind and on paper. When it comes to actually doing something new, and this project is the ultimate example of that, you really have no idea how things are going to turn out. And you certainly don't want to waste time, particularly for somebody who's as talented and smart and motivated as Ari is. So, it's been a relief, honestly, seeing over the last 5-6 years, whatever it's been, I lost track. That's also a typical PI thing, seeing her turn this, nothing really into something, and not only something, but I think one of the things that I'm proudest of having achieved in my time as a PI, because she's really taken Danianella, which at the time that she started was just a fish from Southeast Asia that people didn't even particularly have an interest in from the standpoint of raising them for fun and as a hobby, into a real working neuroscience model that not only is something where we're now confident that we can do additional work in, but is also a system that a lot of other labs are interested been working with as well, to the extent that now I think Ari has probably sent embryos of these fish to 30 or 40 labs within the United States. Which is to say, it all worked. And the reason that it worked is because Ari is a really exceptional scientist and quite good at grinding away and making things happen that are difficult, I think, for most people to do. I mean, I have to say that I'm lucky that Ari took this project on for a number of reasons, not least of which is that a lot of folks probably would have crashed and burned on it. I don't know that I have the ability necessarily to tell the difference between having seen them do work, put the effort into a project, but I will say that I was pretty optimistic that Ari had what it took from the very beginning, and for sure she did. So, I'm not going to belabor it too much more than that. Ari's work speaks for itself. I just want to give you all kind of a more direct sense of like the drama that was involved in the history of this work. And also just highlight for you what a joy it's been to have Ari in the lab. I think the combination of her being the great person that she is and the great scientist that she is has really made this one of her most rewarding experiences in her professional career. And I'm really excited for her to get the chance to tell you all about it.
Can you guys hear me fine? Yeah, perfect. Hopefully in the Zoom, you can also hear me fine. But yeah, thank you. Thank you so much, Adam, for that introduction. It was really nice. And yeah, I'll be happy to tell you guys about my project and how I have gone on to develop this little guy into a new neuroscience model for social reward studies. So let me, sorry. Okay, perfect. So first of all, so socialization is an essential component of for health and survival, not only across human beings, but also across a variety of species, including mammals such as mice or other mammals such as mice and fish. And there are several aspects of these behaviors that are required to maintain it. So social bonds are formed via social recognition, which is the ability to recognize the members of the same species, and social reward, which is the ability to find these interactions rewarding. However, despite the importance of these physiological phenomena, the neural mechanisms that are underlying these behaviors are incompletely understood. So what specific neural mechanisms are required? And in our lab, we have been interested on studying neuropeptide oxytocin, since it is a non-modulator of a variety of behaviors, but specifically social behaviors. And it's really interesting since oxytocin is only a 9 amino acid peptide, but it has a variety of functions, and it is evolutionarily conserved across a variety of species. It is produced deep within the hypothalamus, and it is released in two ways. One is via the pituitary, where it is released as a hormone, but more interestingly and important for us, it is released via the central nervous system via a variety of projections, where it does a variety of behavioral functions. And so we are interested specifically on how these neurons, oxytocin neurons that project to different regions of the central nervous system regulate such complex behaviors such as social preference and social reward. So how can we do this? How can we study what the neural correlates or what the specific mechanisms of oxytocin are in social preference and reward? And this question is a little bit more complicated than it might seem. So what we and several labs use traditionally is the larval zebrafish, which up until 7 days post-fertilization, it remains completely transparent, which is very useful as it allows us to visualize the entirety of the brain of this fish from and all of its neurons at a really high resolution. So this is just a zebrafish brain labeled with a pan-neuronal, so labeling all the neurons, GFP marker, so it labels a green fluorescent protein. And so you can see here, you can see very clearly really delineated cell bodies and really specific cell brain regions. And another important characteristic of the larval zebrafish that's useful is that it is genetically tractable, meaning that you can insert a variety of different tools for genetic manipulation, such as the ability to visualize neurons with a green fluorescent protein, or manipulate neurons with a channel rhodopsin, or visualize its activity with a calcium indicator. And so because of this, you might think, well, the zebrafish, then we have it solved. We can just label the oxytocin neurons and visualize the fish doing this certain amount of behavior and see what happens there. In addition, so adult zebrafish, when the larval zebrafish grow up, they exhibit a variety of mature social and learning behaviors. So for instance, they have a really stereotyped courtship behavior, and they also are able to undergo reward learning. So in this case, it is based on food, but in this experiment, they basically train the fish to identify an area previously unassociated with food, with an appetitive stimulus. So in this case, food, and the fish learned to, when they were trained, spend more time near that area. And so basically, this is just to say that adult zebrafish have these really complex social and learning behaviors. But that is not the case, unfortunately, for the larval zebrafish. So up until their larval stages, when they are transparent and really easy for these imaging manipulations that I mentioned, the fish, unfortunately, does not have a really mature behavioral repertoire. Most of it is very instinctive in nature. And these behaviors do not fully mature up until when the fish undergoes this stage of metamorphosis, which hopefully you can appreciate here that a really dramatic change goes in this at these stages, where the fish starts developing scales, starts developing pigmentation, starts growing dramatically in size. And so by the point they reach sexual maturity as an adult, they do develop these complex social and learning behaviors, but they lose the transparent characteristic that made them an ideal model in the first place. So then how can we bridge this gap then? How can we obtain the advantages of the larval zebrafish regarding transparency, imaging advantages, and the advantages of the adults regarding their behavioral complexity and maturity? And so this is where my little fish comes in. So I want to introduce you to Danionella cerebrum. And as you can hopefully already appreciate here, this is an adult Danionella. And if you remember the previous slide where you saw an adult zebrafish, you will be able to appreciate that the adult Danionella retains some larval features, in the sense that it remains optically transparent, and it retains a really small size. It was considered one of the smallest vertebrates up until very recently. And so because of this, it was a really interesting idea to try to adapt this adult fish to the tools that we are using for the larval zebrafish. And to further convince you about these differences, so here on the top, we have a larval Danionella cerebrum at 24 hours, and here we have a larval zebrafish. And so you can see that at these stages, there's really not that much difference, other than the larval zebrafish has a little bit more pigmentation already. But when you look at the adult, you can see here that the adult Danionella is way more transparent. It is smaller in size, while the zebrafish, you compare it, it's bigger, it has scales, it has an ossified skull. And really, while the adult zebrafish loses all of these larval characteristics, the Danionella still retains them, or retains a lot of them. And also Danionella has a cartilaginous skull, so it means that it's not ossified, which makes it, increases its potential for imaging deep within the brain. So here, this is an adult Danionella that is stained with bone in red, with ulcer in red, and the cartilage is stained with Alcian blue. And hopefully you can see here that the top of the head is completely blue, which means that the skull is mostly cartilaginous. So again, this is a more larval feature, but you can see here that would increase the advantages for imaging techniques. Another really important thing is that Danionella is the closest relative of zebrafish. There is less than 1 million years of evolution separating these two species. And so there are several advantages to this. The first is that because they're so close, there was a really big hope that the tools that already are existing for zebrafish would be able to more easily be adapted to Danionella, especially because zebrafish is a really increasingly used model in a lot of neuroscience areas. And so the hope was that it will be able to adapt them. And that hope also was increased because the other fish that were developed as model systems, such as Medaka and Fugu, are more distantly related from zebrafish, and they were able to be developed as their own model systems by incorporating the zebrafish repertoire, behavioral repertoire, and molecular repertoire. So this is where I started with my project, just going on to validate Danionella as an immortal organism, since it was not, as Adam mentioned, it was not previously used as a laboratory model. So I had to start from seeing if this was even feasible. And so I did this with a variety of goals. The first one was to demonstrate that Danionella can be raised and maintained in laboratory conditions, because we didn't even know that part. It could be that they might not be able to tolerate the same conditions as zebrafish, for instance. The second was to establish that Danionella was a genetically tractable model, just as is the case for zebrafish, so that we were able to do genetic manipulations of a variety of different enhancers and tools that we would like to be using. The third was to demonstrate the potential for non-invasive brain imaging in Danionella. So that way we would be able to visualize deep within the brain of this fish during adult stages when it's performing really complex and mature behaviors. And the fourth one would be to determine whether we can, whether Danionella cerebrum displays robust and particularly quantifiable social reward behaviors, since then we could bridge all of these goals together, or doing a variety of experiments. So for the first thing, so as Adam mentioned, I started seeing if this fish could breed and thrive in zebrafish conditions from ground zero. So thanks to our zebrafish facility, they allowed me to start some colonies in there. And these are my first 2 tanks of Danionella that were originally even in their own separate space of the fish facility. And I had around, in this case, 40 fish per tank. And I had only these 2 tanks to start with. I didn't even know if they would be able to breed, and that was even a little bit of a complication. But now, actually, I have around 15 tanks of wild-type fish, not even counting my genetically modified lines, which I am kind of spoiling a little bit the rest of the talk. But so this is just to show that the fish can indeed thrive in zebrafish conditions. So the second is, well, I wanted to establish Danionella as a genetically tractable model. And so Danionella, I also was able to show that they can readily breed, just like zebrafish do, via external fertilization, which means that we can obtain embryos at a single cell stage. And we can then leverage this to inject a variety of different plasmids with specific promoters that would guide expression into a specific subset of a cell or a neuron that we're interested in, and expressing a particular indicator in those cells. And in order to make them have a stable transmission, derive tools that are commonly used for zebrafish, such as the Toll-2 transposase mediated transgenesis, that would just basically make the expression be stable. throughout generations, so inheritable. And so it would basically be just injecting this plasmid into the single cell stage of the embryo, allow it to grow and see, where it would express. And I was able to do this. And one thing that I want to also highlight here is that I was able to target specific gene expression in Danionella using zebrafish-derived enhancers. So what this means is that when I started this project, we didn't have the genome of the Danionella sequenced. So instead of trying to figure out what specific gene or enhancer would be able to drive expression of oxytocin neurons in Danionella or of specific subsets of cells, we just took the enhancers that we knew worked from zebrafish or derived from the zebrafish genome, injected those same ones into Danionella and saw if they would express there. So this wasn't derived from the Danionella, but from the zebrafish genome. And the beauty of how closely related these two species are is that the genome is so closely related too that this actually was successful. So in this case, I injected a pan-neuronal, so expressing in every neuron enhancer, expressing green fluorescent protein into Danionella. So this is the Danionella larvae. The head is on the top and the tail is on the bottom here. And so this is a sparsely labeled line. So it's just it's not a stable transgenic line, which is why it's not entirely labeled. But you can still see here how there are neurons in a lot of different brain regions. So for instance, these are some neurons in the optic tectum. These are some neurons in the hindbrain. And so this is just to show that this was successfully performed. Yeah, so I also was able to generate stable lines, which means that they are inheritable across generations. And so this is an F2 line. So it's the grand fish of the original line. And you can see here, so this is a line expressing a pan-neuronal calcium indicator. And you can see this is just a video from top to bottom, which, is displaying. So you can just see, this is just to show that there's neurons labeled all throughout the brain. Here in the tectum, there were some in the hindbrain earlier on, and these are inheritable. So this fish was not injected, and they are still being expressed reliably. And this was not only limited to a pan-neuronal expression, so to expressing in every neuron. I was also able to do it, limiting it to specific cell populations. So in this case, I injected an MNX enhancer, which expresses in a subset of motor neurons. And so here is expressing green fluorescent protein in those subsets of neurons near the spinal cord. And here, this is an enhancer for oxytocin, which again, this will be more relevant later, expressing green fluorescent protein in Danionella. And I will go into detail a bit more later, but just trust me that the expression pattern and anatomical location of these neurons is very similar to what is seen in the zebrafish. So that also kind of shows that there is an evolutionary conservation between oxytocin and in both species. And this was not limited to neurons. I also was able to generate expression in a variety of different tissues. This is just some examples. This is some sparsely labeled line that is labeling the cells in the heart. So with green fluorescent protein, so you can see a couple of them here. And I was also able to incorporate some tools that are commonly used in the zebrafish onto the Danionella. So for instance, in this case, I incorporated a commonly used system, the GAL4 UAS system, which is used across a variety of species, to, in this case, express a red-shifted channel rhodopsin into a pan-neuronally, but in this case, it is just shown here near the spinal cord of the fish. So this is just to show that I was able to genetically manipulate Danionella with a variety of different zebrafish-derived enhancers at an anatomically reliable position. So the third thing that I wanted to do was to demonstrate the potential for non-invasive deep brain imaging in Danionella. And so the first thing that we went ahead to do was to inject a fluorescent red dye into the vasculature of the Danionella. So in this case, the heart and the vasculature is labeled in red. You can see it here. This is an adult Danionella. And so we then put that Danionella under the 2-photon microscope. And basically, all we wanted to see was whether we will be able to see all throughout the brain without hitting any obstruction, something that hits, that disperses light too much. And so we were able to do this. So to orient you, the head of the fish, the anterior portion is up, and the tail portion or the posterior portion of the fish is down. And here there is a brain seen from the top, or a part of the brain from the top to the bottom of the brain, and at different depths. And you can see, and you can see how it is, you can see them clearly all throughout these different depths. So that just means that, and this is something that would not be able to be seen in adult zebrafish remotely. And even to see anything, you would need to have really invasive perturbations of the brain. So the fact that this is on a live adult Danionella without any additional perturbations it's really amazing. We were also able to show not only that we can visualize all throughout the brain, but that we also can visualize at really deep, at really high resolution in the adult. So in this case, we injected a calcium indicator dye OGD1AM into the optic tectum of the Danionella. And this is just to show that both the cell bodies and the axonal projections were labeled in the adult and were able to be visualized in the 2-photon reliably. And so we also, it has been shown that functional imaging is also possible in Danionella. So another lab that was also starting to work in Danionella, the Jörg Kubitz lab in Germany, were able to demonstrate that functional imaging is also possible in live adults. So this is an image of the ventral proencephalon, and the neurons is a paneuronal line labeling calcium indicator JChem6. So every time there's increases in fluorescence, that would mean that there is an increase in activity. And so in here, this is just showing spontaneous activity of these neurons. And so every increase in fluorescence would be shown as a peak. And here you see that upon a stimuli, in this case, it's an auditory stimulus, there is an increase in fluorescence in these neurons. So this is just to show that functional imaging is possible to be visualized in a live adult Danionella, which is also an important thing to show. So the fourth thing that I went on to show, and that I'm going to delve a little bit more in this talk, is to determine whether adult Danionella display robust and specifically quantifiable social preference and reward behaviors. And the way that I did this was by adapting a paradigm that was already developed in zebrafish. So the way that it was done was I used a U-shaped arena with a transparent barrier on the edges of the arena. Behind one, a Danionella was placed, or a same species fish, a conspecific. Behind the other one was a neutral stimulus, which is a marble. And then we placed an observer fish, or a Danionella that was allowed to swim from one side to the other. And we basically just saw whether the fish spent more time near its conspecific versus a neutral stimulus, and that would be an indicator of social preference. And then to see if social reinforced learning was present, we projected a pattern on both sides of the arena. And it was a distinct pattern on either side. And so we expected that when the fish was removed, the observer fish would still learn to associate that pattern with the presence of a positive stimulus, in this case, a socially rewarding stimulus, and it would spend time near that pattern in contrast to the other side. And I'm going to go over this a little bit more, since I'm going to be talking about it a lot during this talk. So the way that this paradigm worked was, so during baseline, I just allowed the fish to spend in the arena for 15 minutes just to acclimate. And then I recorded the movement of the fish from one side to the other for 15 minutes. Then during the training phase, I added a conspecific or same species Damianella, a neutral stimulus on the other side. And then I allowed the fish to acclimate to this new stimulus for 30 minutes. And then I recorded the behavior of whether it spent more time near one side or the other for 15 minutes. And during the testing phase, I removed both stimulus and I saw if the fish would spend now more time near the social pattern versus the pattern that was associated with the neutral stimulus. The way that this was quantified was that the area near the conspecific fish or the social area was the number of frames the fish spent on this area were recorded, and they were subtracted from, they were added plus the total amount of frames that the fish spent on the non-social side of the arena, and then the total amount of frames the fish spent from one side and the other side. So it's amount of frames in the social area minus the amount of frames in the non-social area divided by the total amount of frames in both areas. And this is what would be a social preference index. And all you need to know is that a score of 0 means that the fish has no preference for either side. A score of 1 means that the fish spends more time near the social area. And a score of -1 means the fish spend more time near the non-social area. But in order to normalize, I also went and normalized it over the baseline, which means that instead the fish could have every fish that was tested could have an innate preference for some reason over one side or the other. So I couldn't just test whether inherently the fish prefers one side or the other, because that would skew the results. So instead, I normalized every behavior over the baseline so that the SPIs of the training and the testing phases were subtracted minus the SPI of the baseline phase, which gave me a normalized social preference index, or NSPI. And here it's a score of 0 would mean for these two phases that there is no shift from baseline, meaning the fish spent swimming in the same general area in comparison from the baseline to these phases. A score above 0 would mean that the fish during training and testing phases spend more time near the social area compared to the baseline. And a score below 0 would mean that it would spend more time near the non-social area. So really, all you need to know for the next figures that I show is just that 2 is that it's more, prefer more the social stimulus, or not 2, but above 0. And below 0 is that it prefers the non-social stimulus, or at least it's avoiding the social one. And 0 is no shift from baseline. And this is just a video to kind of show how this looks. So this is just during the training phase, and you can see here. So here you have the same species or conspecific fish here. Here we have the subject or observer fish. And in this case, you can see that it's interested on this as opposed to the other side. And here during the testing phase, you can see that the fish stays the majority of its time near now the side. And you cannot see it here because it's just infrared illuminated, but the pattern is actually shining on one side over the other. So it's spending more time near the social pattern in this case. And so, I'm going to have this just to kind of guide you when you see my figures from now on. But again, above 0 means social preference or social reward depending, and above 0 it's the opposite. And 0 is no change from baseline. So you can see here that each dot represents a different fish. And you can see here that the fish, both in the training phase, which would mean that there is social preference, was significantly spend more time near the social area versus in comparison to baseline. And on the testing phase, they also preferred to spend more time near the social area, despite the conspecific being removed at that point, which means that it was reinforced by the social pattern and remained in that area. in order to further show the reinforcement aspect of this behavioral paradigm, I modified this behavior paradigm slightly, in which the baseline and the training phases were still the same, but the testing phases, the pattern was shifted from one side to the other every 5 minutes. And so with the idea that if the fish was reinforced, positively reinforced by this social pattern, it would switch sides alongside the pattern when it was switched. And so in this sample trace, you can see that this is the case. So in the baseline, it's kind of swimming a bit of all over the place. During the training, it spends time near the Pacific fish. And during the testing phase, when the pattern is on the same side, it stays there, then it switches and it goes to the other side, then it switches again and it goes to the original side. But to show a more quantitative way to represent this, so this is the normalized social preference index only of the testing phases. So this is the testing one. So this one where the pattern is still on the original social side, and you can see as expected, the fish, all of them spend more time near the social area in comparison to baseline. When the pattern was switched to the other side, you can see that this longer present, it was removed. And this is because even though some fish still stayed on that side, other fish seemed to spend now more time in the opposite side, which is where the pattern was switched. So there seems to be at least an effect of this pattern on the behavior of the fish after training. And then when the pattern was switched back again in the testing 3, you can see that again all of the fish combined had significantly above SPI in comparison to baseline. Another thing that was shown was that social preference and reinforcement is actually long-lasting. So in order to show this, the fish underwent the same behavioral paradigm at first, but then the Daniela was taken out and isolated for 30 minutes. Then it was put back in the tank with the exact same conditions as it was originally reinforced at. Then it was removed for another 30 minutes, put back in the tank for 15 minutes. And we recorded whether the fish still spent more time near the area with that was socially reinforced at the beginning, and if it still retained that memory, that reinforced memory. And the answer was that in fact was correct. So not only during training and testing, where the SPI is significantly above baseline, but also during the testing after 30 minutes of being isolated and after 90 minutes. And interestingly, when doing this, the same paradigm, but that now isolating the fish for 24 hours rather than just 90 minutes, and then putting the fish back in, we still saw that there was a significant an NSPI of the testing phase significantly, or of all the phases significantly above baseline. So this just means that social preference and reinforcement are long-lasting in this fish, which is very interesting. Another interesting thing that was found was that social preference in Danionella seems to be species-specific in a context-dependent manner. So in this case, what I did was I put Danionella and size-matched zebrafish on either side of the arena. So what I mean by size-matched means that I grab a juvenile zebrafish that is around the same size as Danionella, so that it didn't, the size wasn't a contributing factor on this behavior. And I saw that when challenged with either of these fish, Danionella still preferred to spend more time near its own species as compared to zebrafish, which means that there is a species-specific preference. However, when putting a zebrafish, a size-matched zebrafish on one side and a neutral stimulus on the other, the Danionella preferred to spend more time near the zebrafish in comparison to the marble. So even though there seems to be a species preference when compared between the two, when only putting a zebrafish versus a neutral stimulus, the Danionella would prefer the zebrafish even though it's not its same species. So what I concluded here is that this conspecific recognition seems to be context dependent. And I will go on as to why I have concluded this happens later in my talk. So stay tuned for that. But for now, so what I've showed you is that Daniel-Ella duos display robust social preference and social reward behaviors. And so the next thing that I wanted to ask, especially based on those zebrafish results, is what sensory modalities may be that are guiding the conspecific recognition in Daniel-Ella. So in order to recognize a member of the same species, we rely on a variety of senses. It could be vision, olfaction, something to know that what we're seeing is a species that we care about. And so I wanted to figure out what that was, and then just kind of keep it on the back of your mind. I afterwards went on to ask what the role of oxytocin is on this phenomenon. But first, we're going to focus on the first, my first question. So my first idea was to test what the influence of visual cues would be on species-specific recognition. And this is because zebrafish are driven by visual cues in their social preference behaviors. So in this example, this is an experiment where a zebrafish that is striped, so it's the wild type version of the zebrafish, was challenged in this arena that is impermeable to olfactory cues, so no olfactory cues can go from one side to the other, between a group of fish that was also striped and a group of fish that is mutated to a nacre fish, so it has a white coloration that you see here. In this case, this fish was actually raised with these striped fish. And so because of that, the fish using only those visual cues and visual pattern distinction, preferred to spend more time near the striped fish as opposed to the white nacre fish. But in other experiments of the same group, this was depending on who the fish was raised with. But regardless, this showed that just the fish used pattern indication and visual cues to select who to spend time with. And so I went on to see if that would also be the case in Daniel-Ella. And so the way I did this is, so the previous arena that I showed you was actually not impermeable to olfactory cues. So olfactory cues could have influenced the behavior that I saw there. So what I did is that I grabbed my original arena and I sealed the glass slides to the bottom and the sides of the arena so that no water could leak from the area where the conspecific fish would be located to the other side. And this is just a visual representation of that. So I just added some red dye to just show that water would not go through. When doing the experiment using this type of arena, I saw that, and using the same paradigm I showed you, where I put a Daniel-Ella on one side and a neutral stimulus on the other, the Daniel-Ella preferred to spend time with the Daniel-Ella still, as opposed to the marbles. So even when the olfactory component was eliminated, the Daniel-Ella still seemed to have both social preference and socially mediated reinforcement of the conspecific. But interestingly, when putting a zebrafish on one side and a Daniel-Ella on the other in this olfactory impermeable arena, the effect that I showed you later, where the Daniel-Ella preferred its own species, was not present here. So here you can see that it just was not significantly above baseline, and it preferred to spend equally amount of time between one and the other. So this indicates that even though visual cues are important for overall social preference and reinforcement, they do not seem to be as prevalent for species-specific recognition. So this is summarized in here. But before testing other cues, I wanted to further understand the influence of visual cues into social preference. And so here, to do this, I used an experimental paradigm where I virtually projected a Daniel-Ella into the arena and a neutral stimulus on the other. And so this was just to mitigate other potential factors of having a live fish on one side of the arena. So in this case, it's just a video projected on either side. And the paradigm was pretty similar to the ones I have shown you. The only difference is that I didn't use the testing phase just due to some constraints with the projector. But so this is just a video to show you the what was shown in the social side. So you can see it's just a Daniela swimming around, doing all of its normal behaviors. And when doing this and challenging the one side with the with this video and the neutral video on the other, the Daniela, the swimming Daniela did prefer to spend more time near the video as opposed to the non-social video. So again, it kind of shows that visual cues alone seem to be driving the social preference. I then went to look at more specific characteristics of visual cues that might be driving social preference. And so in this case, in the social side, I projected a static image of a Daniela. So this is just a static fish. It's not swimming around naturally. It's just what you see here is what was projected. And on the non-social side, it was an image that was luminance matched to the social side, so that light wasn't a confounding factor that could drive the behavior of the fish. And when using this static image, the swimming Daniela still prefer to spend more time near the static Daniela versus the neutral stimulus. Then I went to see if features of Daniela played any role on this social preference. So in this case, on the social side, I put a picture of a Daniela, and on the other side, a fish blurb that doesn't have any features but has the exact similar size and shape-ish on the other side. And interestingly, in this case, Daniela did not prefer to spend more time near either the social or the non-social side. So specific features did not seem to be a driving factor for social preference. It was just, at least in this experiment, the rough shape of the fish. So this again reinforces the idea that visual cues do seem to be a strong driver of non-species specific social preference in Daniela. So it does drive social preference, but not in a really species specific manner. And to further show this, when comparing the paradigm where I had the mixed cues, so where it wasn't olfactory impermeable, versus the ones where it was either olfactory impermeable or just a virtual projection, the SPI was, for all of these fish, was significantly higher in the combined cues paradigm as compared to the visual only, either the reel or the video. So that means that there seems to be an additional cue in this combined cue setup that might be further driving and further reinforcing these behaviors in the Daniela. And so my logical follow-up was whether olfactory cues may be playing this role. And this is due to, first of all, the arena that I was used. The only thing that I changed in the olfactory impermeable arena was the access of olfactory cues. The second thing is that Daniela actually inhabit, this is their natural environment. And as you can probably guess from this, it is pretty murky and pretty muddy, and the visibility in these waters may be impaired. So Daniela may rely on other senses, for example, olfaction, to identify conspecifics. And this is also, this has been shown in other fish papers that limited visibility may enhance other sensory modalities in other fish. So in order to test this, I used these, it's a modified arena than the one I showed you. It's a rectangular arena where I have a semi-permeable mesh on either side of the arena, and I have the observer fish in the middle. On one side, I added a social olfactory cue, which basically is I added 25 fish on a 500 ml tank or milliliter tank. Then I waited for 12 hours and I took the water off those fish compound and I put it in here. And in the other side, I put a neutral olfactory cue, which in this case, it's just freshly made fish water. So literally just knew what the recipe for making the fish water in our facility was. I made it and I put it here. So it wasn't even taken from the facility, so to not expose the fish to other fish odors or something. So it was completely neutral. And the phases were pretty similar again. In this case, it would just be baseline, and in this case, exposure, because they're exposed to these new olfactory cues. And the only difference here is that the recording was a little bit shorter for 6 minutes due to the changes in the arena. But other than that, everything else was similar to what I have shown you. So in this case, so I placed a Danionella derived water versus a system water. And I saw that based on these olfactory cues alone, Danionella do prefer to spend more time near these conspecific derived or Danionella derived water versus the neutral water. So olfactory cues does seem to be driving this social preference in a really robust way. And interestingly, excitingly, when I put and zebrafish derived olfactory cues on the other, Danionella preferred the Danionella derived cues as opposed to the zebrafish, which is, as you may remember, this was not the case with the visual only cues, in which case it didn't prefer either side. But here, it does seem to have a robust effect with the olfactory cues, meaning that olfaction may have a more robust effect for social driven recognition of the same species. But to further convince you, hopefully, if I put a zebrafish derived water on one side and system water on the other, Danionella does not seem to prefer one side over the other. So it's not different. The SPI is not different from baseline. So this, again, further confirms that it's not just that there's a presence of a different cue or anything. There does seem to be something derived from the conspecific olfaction or conspecific olfactory cues that seems to drive this behavior. So again, just to kind of summarize all of this that I have told you behaviorally, I have not only shown that Danionella displays robust preference and reward behaviors, but I also showed you that for social preference and reward, visual olfactory cues seem to be sufficient, but olfactory cues specifically seem to be sufficient for conspecific recognition. So now we're going to move to my second question, which is whether oxytocin modulates social preference and reward in Danionella. So coming back to my introduction to oxytocin earlier in my talk. So as I mentioned earlier, when I used my oxytocin enhancer in Danionella to show to label the oxytocin neurons in the larvae, I was able to see that it was really anatomically similar to that of zebrafish, and this is not as surprising since oxytocin is a highly conserved system, but it is really nice to just confirm it. So here you can see that the neurons are constructed to two specific clusters in the hypothalamus, the preoptic area and the posterior tuberculum, in both fish. So in the left here is the Danionella larvae. In the right here is the zebrafish larvae with the same enhancer expressing in the oxytocin neurons. And another thing that is not shown here, but for both of them, you can see projections into the pituitary as well. So that's just a way to show the anatomical conservation of these in these two species. I also showed that the oxytocin gene and peptide are highly conserved between Danionella and zebrafish. So by this point, when I looked at this, the Judkowitz group in Germany were able to sequence the entire genome of Danionella. So based on that, I was able to get the sequence of oxytocin and compare it to that of zebrafish, which is a really easily obtained sequence. And so by doing that, in this case, it's just a comparison of the amino acid map between the Danionella and zebrafish. And the dashes are all similar between, indicate similarity between Danionella and zebrafish, and the letters is just some differences. But two things that I want to just point out here. One is that the actual mature peptide, which as I mentioned is 9 amino acids, is completely conserved between Danionella and zebrafish. Another thing is just that overall, even though it's hard to visualize here, there's around 80% similarity between both species in this sequence. So based on that, then I went on to do several approaches to see if reducing the signaling or the expression of the oxytocin peptide would reduce social preference and reward in Danionella, especially because this has been shown in zebrafish, but we wanted to just further make sure that this would also be the case in these new fish. The first approach that was used was a pharmacological approach. I did it by using an oxytocin receptor antagonist, L368-899 hydrochloride, which is a competitive antagonist of the oxytocin receptor, which would basically prevent the oxytocin from binding to the receptor and signaling. And so, so I added 10 micromolars of this compound in the arena where the fish would be normally doing behavior, the U-shaped arena I showed you. And I allowed the fish to stay there for 2 hours. And then I moved it to an arena without the antagonist and made it go through the original behaviors that I have shown you. And so when doing this, I showed that in comparison to fish that were treated with DMSO, which is just a control, the fish that were treated with this antagonist had no different social preference or social mediated learning in comparison to baseline, and it was also significantly different from the controls that were that had significant both preference and reward. So this means that the antagonist blocking the oxytocin signaling actually diminishes these behaviors. And this was also the case when repeating this experiment, but doing it in an olfactory only system. The second approach that I did to see if reduced oxytocin signaling would decrease social preference and reward was to generate an oxytocin knockout of Danionella. And the approach that I did was just to leverage the tools of CRISPR-Cas9 that have been developed not only in zebrafish, but in a variety of other species, as all of you here might know. And using that, I would inject both a combination of guide RNAs targeting the oxytocin gene in different locations, because as I mentioned, it was sequenced by that point, so I knew the sequence, and inject it into the single cell stage of the Danionella. And so, as I mentioned, I used this approach where I used a combinatorial setup of 4 guide RNAs, which is actually a pretty recent technique developed in zebrafish, where that would allow me to get mutants that were visibly different at the F0 generations, at the fish that were injected in that generation, not having to wait for further generations to be mature to do these experiments. And so to do this, as I said, I have the guides, and this is just an exon intron map of the oxytocin gene with 3 exons in blue. The actual sequence coding for the mature oxytocin peptide is here in red. So you can see that most of the guides that I targeted were near or in the mature peptide sequence, because that's what I wanted to make sure that it would work. And this is the peptide sequence, and you can see again, this is the mature peptide and how these two guides were targeted to that area so that it could, making sure that it wouldn't actually be functional. And so when looking for oxytocin expression using RT-PCR and comparing the fish that I had injected, my F0 crispins, versus the wild-type uninjected siblings, I saw dramatically reduced expression of the gene in this fish. And so I used these F0 crispins and I ran them through my behavioral setup. And I saw that there was, just as in the pharmacological approach, there was reduced non-significantly different from baseline social preference and social reinforcement in these crispins in comparison to the non-injected siblings. So this is just further to show that oxytocin signaling does seem to be involved in social preference and reward in these species. So as a summary, just to kind of show what I have told you today, what my project has consisted about, and what my contributions of the field have been. One of the most important things is just that I was able to validate Daniela cerebrum as a new systems neuroscience model and show that it has the transgenesis and imaging advantages of the larval zebrafish, but it has the behavioral complexity and maturity of the adult, and both of these things can be combined to do a variety of different experimental questions, not only limited to social behavior. And I also was glad to see that I could, just as in that new zebrafish paper that came out, I was able to generate mutant lines within a single generation using this combinatorial guide RNA. I could obtain crispins that could be, where expression could be easily be visualized as being reduced, and with behavioral phenotypes that were different at a single generation, which is useful for Dionella because I forgot to mention that they don't lay as much as zebrafish do, and it's a little bit trickier to get them to lay that many embryos. So this is a really neat thing to show, especially for Dionella. That was a big contribution. Another thing, just as I showed you for a great majority of the stuff, was that Dionella has this quantifiable social approach on learning behaviors. So with this, this has shown that tools that are available in zebrafish, also not only the molecular ones, but also the behavioral ones, can be easily adapted and quantified in the system. And also just to further understand the neural underpinnings of these two really important behaviors, they could be leveraged with this new system. Looking specifically at a little bit more of an ethological discovery, I found that olfactory cues seem to be particularly important for species-specific recognition in Dionella, and it seemed particularly interesting due to the environment in which they usually live, and it would lead to really interesting experiments seeing whether it is just a sensitivity to social cues or whether it could be sensitivity to other olfactory cues. So that is something that could definitely be explored now in the system for olfaction studies. Finally, I showed that oxytocin modulates social preference and reward in Dionella. And so this knowledge could lead to future experiments in which the exact neural dynamics, activity dynamics could be assessed using a variety of cues. For example, using a social cue, putting a fish under the two-photon microscope, seeing what particular activity dynamics of these neurons might be upon this stimulation or this exposure. And this also overall just gives insight on the role of oxytocin in social preference and reward. It seems that it's conserved in Dionella as expected, but it still gives insight to that. And of course, it gives groundwork for future oxytocinergic studies in this system as well. And so I would like to thank all of you for coming today to my defense, but I also want to thank Adam for being such an amazing PI and for honestly, it has been the best mentor I could have asked for in grad school. And so I've been really happy all these years working in his lab. And so, yeah, so it's been amazing. I also want to thank all of my committee members for being also so amazing all these years and for offering so much support throughout these years as well. And obviously this also goes to Adam, both in scientific and non-scientific matters of my academic development. It's been great. And a special mention of thanks to Rich, who is my unofficial sixth committee member, since we are basically sibling labs at this point, and he has been extremely involved in my project as well. So I wanted to give a special thanks, since he also has been a great part of my development as a scientist and of the development of this project. And of course, I want to thank my lab as well, both past members and present members, which I know here includes the Dorsky lab, but as I say, it's a sibling lab. So I want to thank you guys as well for just such a collaborative environment and such just able to help with anything and being just a great lab atmosphere to be all of these years. And I especially want to thank some current members of my lab, particularly Emily, Ariana, and Paula. You have been amazing and just, again, super collaborative and a great atmosphere. You have created a great atmosphere in this lab, so, and also great friends as well. And also a really, really big thanks to Jacob, who was an undergrad in my lab. He came as a sophomore, and you might have seen his name in a lot of the behavioral experiments that I mentioned in this talk. He was a great help for running all of them, and it's been a pleasure to see him grow as a scientist, and now he's going to attend grad school as well this year. So it's just been great to be having him, and this project could not be completely been made without him. And of course, I want to thank the neuroscience program, both Tracy and Nicole, for just being so helpful all these years. Obviously, the neurobiology department, Monica has been a great director of the department. It's been great, and Emily Harris. Also the SAR, who allowed me to just put a random new fish in the facility and had to deal with all of the things that entailed. Both Maureen, Sharon, and Carrie for just your patience at all of my requests. And I also want to thank, so as an international student, the milestones that one has to go through to be studying here are sometimes a little bit greater. And I really couldn't be here right now if it wasn't for my high school principals, Mr. David and Gloria Bender. Because of them, I got the opportunity to study in the US since my last year of high school in Phillips Exeter, and they have been with me all throughout these years, just supporting me, just showing how life in the US is like, supporting me with applications, with advice, a lot of things. So I really want to just give a big thanks for all of their help. And I know Mr. Bender is in Zoom right now, so thank you so much, and the Harkness Institute as well. I also want to thank my college advisor, Greg Tepper, which I know he's also in the Zoom. I saw him earlier. He also was a great asset and a great developer of my scientific career. Allowed me to find the mythical internships that are a little bit hard to find as an international student, and just always supporting, and I'm just a great teacher and mentor. So I want to just give a big thanks to him. And I also want to thank my friends, especially there's some friends, and maybe it's because I'm an only child, but there's some friends that are more siblings than friends to me. And for me, it's these four, Kyle, Emily, Tom, and Hallie. They're amazing friends. I've known them since college, and honestly, they have been just great, and I couldn't have wished for better friends. And to show you an example of what kind of friendship this is, Hallie currently is in Disney World, and instead of riding a roller coaster, she went to an office or something to watch a PhD defense. So this, yeah, so this is what this kind of friendship is like. So that's why I'm like really helpful for having them as friends. Really thankful for all of them. And I also want to thank Peter Wynn, who has Dr. Peter Wynn, who there's some people that in grad school just help you go through all of the different difficulties that arise along the years. And Peter was an amazing emotional support through, as we both went through grad school together, was a really great help in my qualifying exams, but just he's always really supportive and really helpful in everything. So I wanted to give him a really special thanks because he's just a great guy and a great friend. I also want to thank my college roommate and really good friend, Bridget, who I think of all of the quizzes I've ever done, the quiz of which roommate you will get in college was the one that I had aced the most because we are really similar in, she wanted to go to med school, I wanted to go to grad school in college, but now she's a resident in neurology here at the U, of all places, and I'm finishing my PhD in neuroscience. So this is how similar we are. And it has been amazing to both have her as a roommate, this is our first roommate picture, but also have her as a friend now in Utah and just really supportive at all times. And also there's so many friends that have been extremely supportive and extremely helpful to me throughout my life, both past and present. There's too many to put on a collage or to name here, but just know that I am extremely thankful for all of you, like Fer or Erika, and obviously Luciana and Claudia and all the Garcia Crespo family. Thank you so much for all of it. I also want to give thanks to two really special role models in my family. One who unfortunately are no longer present, but I know that they would be really proud. One is my aunt, who she's the first person that I heard what a thesis was, because she was working on her thesis when I was like 6 years old, and she allowed me to use her computer for some reason while she was working on it. And she was like, sorry, I'm writing my thesis right now. You can't use my computer. And I wouldn't let my computer to a 6-year-old now that I've been writing my thesis. I'm really astounded now that she did that. But she finished her thesis. She did graduate. So I didn't do too much damage to her computer, luckily. But she was an amazing aunt. She just was a great role model of, oh, look at what women can achieve. That she was always looking to learn new things, and I know she would be really, really happy and proud to be here. The other one is my grandma, mi abuela Betty. She also was a really great female role model growing up. She was always for education of women. She was really ahead of her time. She also taught me a lot of cultural things. She was a piano concertist, but also just extremely supportive. I know she would be really happy to be here too. So I wanted to mention her because she was just an amazing grandma, and she just was a great role model to have growing up. So. Also to my grandparents, even though again they're also not here currently, but I know that they all would be happy, both from my mom's side and my dad's side. I wouldn't be here also without all of their support. And also, of course, the rest of my family as well. Again, just with my friends, I've been really lucky to having such amazing family members that have been extremely supportive. Again, too many to name, but my Uncle Rich, my cousin Datus, Rodrigo and his family, and a big hello to my Tío Loco, who I know is watching from Argentina, so I want to say hello. But yeah, I had amazing family as well. I want to thank all of them, even if they're not in this collage. And finally, my parents. They have been, I couldn't have wished for better parents, and really they kind of have earned in my sense. They have gone through this PhD journey as closely and intricately as I have been. They know all of my failed experiments, all of my troubles I had, all of my successes, and of course, not only on my PhD, but also in all of my other aspects of my life, starting from birth. You know, they've been amazing parents. I couldn't ask for any better parents, and I words cannot say how happy I am to have them. So, thank you. If you have any questions, or either in the Zoom, if you want to type questions, you can. Yeah. Oh, yeah. So yeah, the site in which the pattern was projected was randomized across trials. So yeah, so I checked for that, and I also just did an average SPI score of all the fish in the different pattern combinations, and I didn't seem to see any bias over one pattern side or the other. Yeah, or color. Yeah, that yeah.
Amazing. Especially for someone who doesn't do anything in neuroscience.
Thank you.
I did kind of want the same line of question. When you switch the sides,
Mhm.
did these start one side the other? Did you keep them in the active? Do you think that?
Yeah, I mean, I acclimated them during the original, so the baseline time. I didn't do it during the switching pattern. I think, I mean, probably it would be interesting to see what happens if they stayed on one pattern for a longer time than 5 minutes and seeing if that would affect it. But I didn't do it during that just because I was afraid. I wanted to do it right away rather than allow them to wait and potentially diminish the reinforcement effect. Thank you. So, I mean, so yeah, so as I mentioned, they didn't seem to have a preference for one side or the other. And I also randomized not only the pattern, but the side in which the social stimulus would be presented. But when looking at the average of all of those experiments, I didn't see there seemed to be a bias for one side or the other. Yeah, but I also randomized them just in case. Yeah. Yeah.
Fantastic talk. I am curious about the time of day that the assays were done. If there's any circadian influence on this.
Yeah, I tried to do it during their normal. So I did it usually after 10 a.m. and I stopped doing it around like 4. So I never did it when it was their dark hours in the SAR, and I didn't do it when it was too close to their initial light phase. And I also had a light and dark phase if I left them to do experiments with theirs. So I don't know because I haven't done it at night or anything, so I don't know if there might be any effect with that, but I didn't seem to see any difference regarding the time of day. They all seem to be, in average, pretty similar. Yeah. I don't see any questions in this room, so just let me know that.
Gonna have Stephanie stage at 1:30.
Yeah, yeah, yeah.
I'm really proud of you. Good job.
Congratulations.
I wish I could say I have to.
That's okay.
You're so sweet for picturing me. Since it's the part where the like.
They like kick everybody out and they like.
Okay.
But if you want to stay, I mean, the thing is not until 1:30, but like most people have stuff.
Sure.
Like, the times you wanna wait, I don't know how.
Where's the stuff?
I don't know yet. So just like I can wait outside with my parents and then we'll figure it out. Yeah.
I think Instagrams.
Yeah. Okay. Yeah. Thank you for coming.
Yeah, of course. That was a great talk.
Oh.
One question, are you gonna get a Daniel Owl?
No.
You're like, oh, sick of them.
Yeah, Adam asked me the same thing. Okay. Okay. Oh.
Very good.
How are you? Good to meet you.
My dad. Yeah.
Good to see you.
Yeah.
Okay. Let me just close it Zoom now.